we have seen most of the basics of science and technology. So we'll quickly start the current affairs related to the things that you have studied. Okay, what is genome sequencing? Yeah, actually we know that in our DNA, that is our genetic material, the genetic information is recorded with the pairing of a few alphabets like A, T, C, G, like that. Okay, so determining the order of that base pairs. Okay, A will be pairing to T, C will be pairing to G. In somebody, like in my genetic material, it will be written as A, T, C, G, A, T, C, G, like that. In your genetic material, it will be A, T, T, like that. It will be written. So that difference make us different. Okay, that difference in arrangement of that base pair make each individual difference. So reading of this genetic information, how this genetic information or base pairing is written in my genome and your genome, we'll open up a cell and in laboratory we'll read that sequencing, that order of that base pairing. That is known as genome sequencing. Okay, so about genome sequencing, for example, if a person is infected with a particular virus, we are taking some samples of cell from within his cheek or from his throat, and we will be sending that to lab. Okay, then we are finding that like it will be having two genomes, one that person's genome, second that virus genome. Okay, then when they are observing that virus RNA, they are coming to see that, okay, ACCAU, like that. Okay, actually there is one difference between the base pairing in DNA and RNA. We told that there are two kinds of genetic material, one is DNA and the other is RNA. In most of the organisms, apart from certain virus, the genetic material will be DNA. Okay, but in viruses in general, that can be RNA as well. So normally in all the organisms where DNA is the genetic material, that base pairing will be done by 4 alphabets, A, T, C and G. Okay, but in the case of virus, in the place of T you can see U. Okay, in the case of viruses where the genetic material is RNA, okay, you can see U, but this U won't be present in DNA. DNA, like in the organisms where DNA is the genetic material. So when seeing this thing, this U or this chemical, okay, then this uracil, when it is observed in the lab, he can identify that there is a new genetic material in this person's sample. Okay, so that means an external organism's presence is identified in this sample when they see this in the laboratory. Okay, then what they'll do is actually they'll be having a record of different types of viruses like genetic information. As I told when I was teaching that basics, in 2003 there was SARS virus. So they already have the genetic order or the order of base pairing of SARS virus. Okay, the order of A, G, C, U, like that arrangement in SARS virus they already know. Now in 2023 they will be comparing this person's RNA, like that virus sample obtained from this person's body that will be compared with the 2003 virus sample. Now they'll cross match, and if all the order is same, that means both are same virus. Okay, so the vaccine which was developed for 2003 SARS could be used now for this virus as well. If the order is different, they need to develop a new vaccine. Okay, that may not be applicable for that particular virus. So this is one application of genome sequencing. So what is a genome sequencing? What is a genome? First of all, it's an organism's complete set of DNA including all of its genes. We are having like lakhs and millions of cells in our body. The entire DNA, every cell is having a DNA. Okay, every cell in our body will be having same DNA, same order of base pair in a single person's body. In every cell there will be same set of DNA, but that will be different from you and me. Okay, but in my body all cell will be having the same order, same base pair arrangement. That's why we told that in crime scenes when we are getting one like hair or one nail sample, we will be able to identify that person if that person's DNA order is already there in the crime database. Will match with the other criminals, will match. Okay, then we'll be identifying that. But in one person's body, that will be same. In every cell will be having the same set of DNA, and the entire set of DNA in total of our lakhs of cells, that is in total is called genome. Okay, each genome contains all the information needed to build and maintain that organism. We told that if we are obtaining one hair from the like Hitler's body, okay, we can recreate that person. Okay, because there are techniques for multiplying the DNA. For example, we are having PCR technique. When we are obtaining one sample of cells from our cheek, in laboratory what they are doing is they are multiplying this. Okay, they are multiplying this genetic sample so that lakhs of DNA can be created from one single copy of DNA. So they can create from one single cell, one teeth or one nail or one hair piece of a dead person or a dead animal. If they are obtaining, they can recreate that organism. Okay, they can create artificial cells and they can make tissues. Now there are devices we will study today, technique is called 3D printing. Okay, 3D printing means if you are giving one input in computer, you are having a particular design. For example, the shape of our heart. Okay, the shape of a human heart, that will be drawn and that will be kept in a developed as a 3D drawing in computer, and we are adding in that computers in place of our normal printer where we are adding ink. In this 3D printer, we will be adding human cells. Okay, human cells that will be added. Now, when we are switching on that printer, these cells will be coming in the shape of a heart, human heart. That can be used in human. That was tried as well. Okay, so these kind of techniques are available. So if a single hair sample from a dead person is available, we can create that entire person using 3D printing even now. Okay, so that is the because all the genetic information needed to build and maintain that organism is available in the genome or DNA of that particular person. In humans, a copy of the entire genome, okay, more than 3 billion DNA base pairs, okay, is contained in all cells that have a nucleus. We told that we are having all the DNA of our body is arranged in a coiled form known as chromosome. Okay, in every cell there are pairs of A, T, C, G, A, T, C, G. Like when we are opening up chromosome, we can see that. Okay, that's known as base pairing. A is paired with T, C is paired with G, again C is paired with G, A is paired with T, like that. So we are having such 3 billion base pairs. Okay, in our cells, 3 billion such base pairs of A, T, C, G, like that. So that is being said here. So genome sequencing is the figuring out of order of these DNA nucleotides. We told that A, T, C, G, etc. These are actually known as base pairs or nucleotides. Its chemical name of these 4 chemicals are known as nucleotides. So they are known as DNA nucleotides, the nucleotides which are present in the DNA or bases. Okay, so determining this order of A, T, C and G in a person is known as genome sequencing. So the same thing is being said here. Then what is genome editing? If in a particular person, in a normal healthy individual, if the order of the DNA base pair is A, T, C, G, like that, if in the place of T, by a genetic mismatch, if it happens to be C, okay, like when we are comparing healthy individuals, normally the base pairing will be in a certain order, but in persons with cerebral palsy or autism kind of people, okay, people are having that kind of genetic disorders like maybe sickle cell anemia, hemophilia, etc. Their base pairing, this order will not be like a healthy person. Okay, in place of T, when in normal healthy person where it is having that AT arrangement, in this kind of people, in place of AT, they will be bonded with maybe A may be bonded with C. Normally, the natural arrangement of a person or a healthy individual's order is A must be paired with T and C must be paired with G. But that order can be different. After AT, it can be again AT, AT, AT like that, then CG, AT like that. But A must be paired with T. What happens if in the place of T, C comes, or in the place of T where there is nothing like that. So genetic mismatch, this kind of issues with the base pairing, that creates problem called genetic disorders. So sickle cell anemia kind of diseases are caused because of this genetic disorder. The people that we see, like people with or children with differential abilities, like they are having these kind of genetic issues. Normally that cannot be treated through some medicines, but that can be cured now because of the technique called gene editing. Okay, if a person, if a single a baby is born, for a father is having this genetic disorder, and it is evident in his body that he is having sickle cell anemia. It is doctor has validated that he is having sickle cell anemia. Now there are high possibilities that when a child is born to this person, that child will carry the sickle cell anemia. Okay. Now what they will do is, they will take the sperm of this father and they will fertilize that with the ovum or egg of that mother, and that embryo will be formed, that first cell of that baby. And the first cell of that baby will be having DNA from the father and DNA from the mother. Okay. Now what they will do is, in that first cell, this first cell will be multiplied to create that person. Finally, that child will be formed by 1 cell become 2, 2 cell becomes 4, 4 cell becomes 8, like that it will be like that. Finally that child will be created. But that very first cell, what they will do is, they will edit the father's DNA. Okay. In father's DNA, there is a genetic mismatch. A was not paired with D. In that single cell, they will correct all the areas where A is not paired with D. They will cut that mistake, and they will add T in place of the mistake. So this is known as gene editing.